Today, we have with us Dr. Ron Piran from the Azrieli Faculty of Medicine, Bar-Ilan's medical school up in Tzfat. Dr. Piran joined Bar-Ilan University after 6 years of postdoc research in California at the Sanford Burnham Presbys Discovery Institute. Dr. Piran began his research career, though, as a doctoral student at Technion's Faculty of Chemistry. And there, he was focusing on computational methods for stem cell development in embryos. However, he's now, and he started with his postdoctoral research, focusing on diabetes, which is what he continues to investigate as a researcher at the Dangoor Center for Personalized Medicine. Dr. Piran, welcome. Thank you for joining us today.
Thank you for having me.
It's really a pleasure to have you here.
It is.
So, Dr. Piran, you're now studying diabetes, which is a pandemic across the entire world. By 2030, there may be close to 440 million people suffering from this disease. Mm-hmm. But even before we get into the details of what you're working on in your lab, maybe it's worth starting back a little bit. What is diabetes exactly?
Sharon, this is a great question because people assume they know diabetes because everybody knows someone with diabetes. So let's take it from the developmental perspective.
Okay.
When we start with a single cell organism, very low in development, evolution, development and everything, the rules are very simple. Mm-hmm. If you have food, eat it. If you eat a lot, you'll grow. If you grow enough, you have to divide. That's it, simple. But when we climb up the evolution ladder, things start to be complicated because, let's say we go to a colony of cells, this rule still applies, and colonies do grow, and some parts of the colony grow faster than the others because maybe they are closer to the current with all the nutrients and stuff, and other parts of the colony is a little further away and food is less accessible, and this is still fine. But when we have a multicellular organism like ourselves, we have developed organs, special organs. Our hands cannot eat, our eyes cannot eat. So if we eat and cells will consume all food that they will have, we will just have a very long digestive system, basically like a worm. Mm-hmm. But if you want arms, sensory organs, things like that.
The brain.
Yeah, exactly. We cannot apply these rules. Mm-hmm. So evolution, God, creation, whatever you want to call it, invented a rule. You cannot take all food that is near you. In order to get the food inside the cell, you have to have something that will open the door, that will open the channel, that will allow food to enter. And this one is called insulin. Mm-hmm. So in order to food get into our cells, we have to meet two terms. One, that there will be food around, mm-hmm. and second, that there will be insulin to open the door to allow food to enter.
To enter the cells.
To enter into the cells. Mm-hmm. So, if we don't have enough insulin, or insulin signaling for some reason is not working properly, then the doors are closed, food cannot enter, and only as a byproduct, food and glucose being a major food component builds up
the
in
sugars,
the blood.
Basically.
Exactly.
Glucose is the blood sugar that
exactly.
we think of.
So the blood sugar rises because it doesn't enter the cells. So elevated blood sugar levels are a byproduct of the disease.
Which is essentially either there's not enough insulin or the system that has the insulin opening the doors, as you say, to the cells to take in the glucose isn't working.
Exactly.
So it floats around in the blood and doesn't get into the cells.
That's right. And this creates two problems. One problem is starvation. Mm-hmm. And many, many years ago, especially in type 1 diabetes, which we may go into details later on, these children were starving. And the other problem is that if sugar builds up in the blood, it creates something that is called osmotic pressure on the blood vessels and dehydrating the cells, absorbing water into the blood, elevating blood pressure, and may later cause kidney disease, neurological complications, eyesight problems, and
heart problems, all kinds of vascular system problems that are the blood system, right? So from what I understand, you're saying the sugar floating around in the blood causes dehydration, which means that the vessels bring in more water. They put pressure on the vessels, and that causes all kinds of issues. In addition, which I never thought about, it's very interesting that we usually think of starvation, or as you kind of hinted at, hunger, an issue of putting food into our body, but it's not just about that. Yes. Obviously you need food, but it's about whether the body can break down the food, put it into the places where it needs to be, in particular the glucose levels. I understand. So,
and another thing, diabetes is not necessarily a disease of blood glucose or sugar. It's just that the sensor that we use, the glucometer, we found a way to link between glucose levels and electric current. So our glucometer checks directly glucose levels, but diabetes patients accumulate other nutrients as well in their blood, including free fatty acid, glycerides, triglycerids, and other nutrients that are also floating in the blood and don't reach.
They can't get into the cells.
Exactly. So diabetes is not only glucose.
It's just that that's what we can test.
Easily, yes.
Easily. And if there's glucose, it usually means, or almost always means, that you have all these other nutrients floating around and not getting into where they need to be. So it's a testing thing as opposed to the only underlying issue.
Exactly.
That makes a lot of sense. So what do you specifically look at in your lab in terms of diabetes?
So type 1 diabetes, what we used to call juvenile diabetes, but this name is slowly being pushed out of the jargon, is a autoimmune disease. And an autoimmune disease is a disease in which the body attacks itself. And I will allow myself to elaborate because...
Please.
So why do we get autoimmune diseases? So we don't really know. But we think that our immune system needs to test and needs to experience and need to learn the self and external invasions like bacteria or other diseases, viruses, whatever. And people, especially kids, who are not exposing enough their immune system and trying it out outside, will make the immune system confused. What I'm explaining to students in class is that let's assume we have these 3-year-old child coming and visit the grandparents. If grandma and grandpa were prepared and they put a few toys on the table, then everything will be fine and the child will play. But if the house is completely clean, everything is tidy and organized, they will get to the china, right? and this is what happens. So if we have these parents, we call them helicopter parents, right? They are hovering around above their children, hand sanitizers in one hand and the
very familiar
wipes, yes, and the pacifier falls into the sandbox or everything, and immediately there is the distraction operation, and then the pacifier is being taken and being cleaned,
sanitized, right?
This is not necessarily good for us,
right?
Our immune system needs to work. If it will not work, it will get to attack the self because this is an active system. So in type 1 diabetes, for some reason, the immune system specifically attack the cells that produce insulin. They're called beta cells. And once they are destroyed, there is nothing to secrete insulin, which will later allow for food entry into the cells.
And the beta cells live in the pancreas?
Yes.
Which is where insulin is produced.
That's right.
So
this is type 1.
Well, just stop for one moment because you're saying something that I've never actually heard before. My understanding, and I assume many people, is that children are born with type 1 diabetes, or people are born with type 1 diabetes, and you're suggesting, or at least I'm hearing from you, that because it's diagnosed as an autoimmune disease, it's possible that there are things that could be done to lower the chances of a child developing it by exposing them to different pathogens or different things that the immune system can fight against.
Yeah, let's not call them pathogens. We don't want people to throw their children into
Bacteria,
the sewer.
Right.
But we do,
external things that they need to.
If a child finds a stick on the ground and puts it in their mouth, you don't sound the alarm just yet.
Okay. You don't have to swab their mouth with alcohol.
Exactly, exactly. Let kids experience nature in the most natural way. And let them experience with food, let them rub things around, touch, put in the mouth, allow this. This is a testing mechanism that we all mammals have to identify and to distinguish self from non-self. This is very critical.
And so introducing the non-self allows the immune system to respond to the non-self as opposed to the self, which is what the autoimmune responses are. And so you were starting to say that part of what happens is that the beta cells are attacked by the immune system, the beta cells from the pancreas that produce the insulin. Is this the area that you're focused on specifically in your lab?
So, yes, but let me allow...
Please, you're right, you only talked about type 1.
Exactly.
Go ahead.
So we have type 2 diabetes, and this is actually, we'll divide it into two diseases. One of them, just obesity, being fat. Being fat, being large, is a problem because, as my colleague says, Dr. Alon Barash says that in every fat person there is a small little human. And the translation of that is that when we are fat, when we are obese, our internal organs don't grow with obesity. They stay the same. And hormones, insulin included, are acting according to concentrations, active concentrations. And concentration is the amount of hormone divided by volume. So when we increase the volume of the person by being fat, that means that more insulin is required to reach the same concentration. And because obesity is a chronic disease, and sorry for saying that it is a disease.
It is, well, you're sorry, it is a disease.
Well, the jury is still out, but I agree with you. We are on the same page.
Well, I actually think it's a good thing because it takes some of the blame off of people.
Definitely.
And it acknowledges that it's not so simple. Don't just, stop eating so much. That's not what obesity is. And we understand that scientifically these days.
That's right. So because it's a chronic disease, these beta cells that secrete insulin work so hard and they never get time to rest. And slowly they become exhausted and die again. So we have another reason for cells to die. The second part of type 2 is what we call insulin resistance. We don't really understand why, but for some reason, many of us will develop insulin resistance. That means that with time, with age, we will need more and more insulin to reach the same biological effect. We don't really understand the mechanism. But the end result is exactly like in the obese people. We need more and more insulin.
And then the beta cells die because they're working so hard?
Exactly. So, and we have other types of diabetes that we will not go into right now, but the end results of all diabetes types is the destruction of beta cells. So only now I'm answering your question, what I'm doing in the lab. So in the lab, we are dealing with finding methods to regenerate those insulin-expressing beta cells.
Which is necessary. I'm glad you laid out the whole thing because it's very clear that whether it's type 1 or type 2, the end result is the destruction of these critical beta cells from the pancreas, or you end up with all the problems that you outlined initially with the free-floating things in the blood and the doors not opening in the cells. And so how are you doing that?
So in that, our path is very unique. So unique that we are finding it hard to explain it. But
try.
I'm coming from a different background, which is formal logic. Formal logic is a branch of pure mathematics, algebra. During my PhD, I have described new math to deal with cellular responses and embryonic development.
Okay.
Using this math, I have forecasted a new pathway that will lead for the development of muscle cells.
In the embryo.
In the embryo, yeah. And we proved that, and I was very proud of that.
Did you prove it mathematically?
Yeah, we started mathematically, and mathematics predicted the existence of the biochemical pathway.
And then you proved it.
And then we proved it experimentally. I was so proud. And
I'm sure.
I wrote this complicated math in the manuscript and so on. And the end, nobody
understood it.
Nobody. Mathematicians didn't understand the biological part.
Even I said to you, practically, Tachlis, tell me, did you actually prove it in the cells?
So it was cited only a handful of times, and people did not realize what was going on. And reading that is extremely tough. So I thought that if we will take this model that I have developed and moved it to a real disease, having real impact on people, maybe people will start paying attention. So we're struggling there. We have formulated the mathematical automata, we call it, or Turing machine is something that the audience might know. Alan Turing.
Yes.
Maybe we'll talk about history a little bit, but he has devised a new, it's not new, but a machine, theoretical machine, that can predict and respond to signals. Now, diabetes is interested in two terms. First of all, it's a horrible disease, and mathematically speaking, there are not only beta cells there. There are many, many cells in the pancreas. All of them secrete something. So the cells are not only responding to signals, like in the embryonic development, but they're also secreting, writing back into the system, requiring the full Turing machine that I just mentioned. So mathematically speaking, this is also very, very interesting. So we have finished compiling all this math together. This was published 2 years ago, and now we're at the end stages of proving that, and currently in mice, we are able to regenerate those beta cells in vivo, meaning in the animal, not taking, cells outside, doing manipulation in the lab, and then reintroducing the cells. No, in the mouse, by giving a treatment, we're able to regenerate those beta cells.
So that wasn't so complicated.
Okay, thank you.
Because you didn't get into the math. If you tried to start with, I was going to say numbers, but your math is probably all letters at this point.
Exactly.
So you bottom lined it for me, and I appreciate it. There was this complex mathematical formula and complicated input, output, but bottom line, you applied it in mice, and it seems to be having the effect that you were hoping for, which is the regeneration of these beta cells. And how far along are you with this?
So we are now confident that in mice, having all the reservations and everything, that in mice this is working.
Wow.
Okay, and the next stages is, first of all, scientifically speaking, it's not enough to show that a drug works. We need to show the mechanism. So this is where we're at right now. We are trying to use different mechanistic studies to show exactly how and where these new regenerated beta cells are coming from. We have great help from Gil Leibovich from the Hebrew University, Hadassah Medical School, that gave us some tools to help examine that. And another thing that we are also pretty concluding right now is the relevance to human cells.
Right, most of us don't care about mice that much.
Yeah, exactly. So we got some primary islets, they're called. So beta cells and the other endocrine cells are in a structure called the islets of Langerhans. Okay, so Langerhans, he looked at the microscope and he recognized these islets. He didn't know what they do, but he was the first one to describe them. And these islets contain beta cells and other endocrine cells, and we got some donations of primary islets, gave the drug to the islets, and showed that indeed the same pathway that we saw in the mouse is also working in the human.
So the pancreas itself is creating?
It's not the pancreas, it's the islets.
Are they near the pancreas?
No,
situated?
they are inside the islet. There is a method. So the pancreas itself is not only endocrine. It's not only producing hormones, it also produces digestive enzymes.
Okay.
Digestive enzymes are a horrible thing to deal with because they digest food, and we, biological samples, are food for the enzymes. So we need to separate this exocrine tissue from the endocrine tissue. So we're left with the islets. We tested the drug that we have on the islets, and the islets respond in a similar way to the response in the mouse.
And the islets are responsible for the original development of the beta cells always?
Yes,
they always come out of there?
Yes.
Okay, so you essentially have proven that you could generate or help the body regenerate from the same place naturally almost the new beta cells. True. Which is very exciting.
Yes, for us, yeah, we're very, very
well, not just for you.
Yeah,
if we're talking
yeah,
about hundreds of millions of people around the world who are struggling with this, and diabetes, anytime you read Dr. Google, what could cause, usually diabetes is listed as one of the causes of every disease out there. It's really the metabolic disease that's so problematic. So this really feels like it can move us forward. What does the process look like? So now you've moved from mouse to cell.
Yes.
And what happens next?
So now this is a good question. There is a question if we do need to go forward to bigger animals. Sorry for saying that.
Well, we're bigger than mice. You can say that.
Okay.
Not insulting.
Okay. And there might be a chance that we will get an approval to go directly to patients.
Wow.
To test. We're now checking the field. What are the next requirements? There is a chance that we will be able to move directly to patients and test the drug.
No side effects so far.
Yes, in the limitations that we can, and that depends on the delivery system. So the drug is being administered by an infusion.
Okay.
The concept of infusion is complicated to explain to a mouse. Okay, so they cut the pipe. So you cannot give an infusion to a mouse. The results of that is that we have to put the infusion bag, or these are called osmotic pumps. We put them under the skin of the mouse. So we cannot hook and unhook the mouse from the infusion, and we cannot test in the mouse what will happen after we stop the drug. So there is a question: is it a lifelong requirement to continue and get the drug throughout your life, which is very unpleasant? But considering the alternatives that we have now, which is continuous infusion of insulin,
correct,
and we didn't talk about hyper and hypoglycemia. Glycemia is glucose levels. Hypoglycemia is also a major problem of diabetics.
Right, when the blood levels drop,
exactly,
and they could die.
Exactly,
from that.
That's the most dangerous thing. So comparing it to insulin, this is not so horrible. So maybe some of them already carry a pump of insulin.
So you could, instead of insulin, you can do this.
So that may be, or there is a possibility, hopefully, it will be great if that's the case, that we can just, after beta cells were regenerated, we can pull the whole system out, and there is no need for the pump.
So right now you don't yet know whether once they're generated they continue, or whether there's going to be a constant need to infuse, or whether you'll generate them, those will die off, and then they'll have to go for another infusion at some period of time. You don't have that tested
especially
yet.
in the case of autoimmunity.
Right.
Because we did not deal with the autoimmune
with the underlying problem that caused the initial killing of these beta cells.
Exactly.
Although it seems like in diabetes, we don't know the mechanism, but there may be something that's causing it also that...
So we have a reason to be optimistic.
Yes.
Because there is a mouse strain that is called non-obese diabetic, NOD. If you'll Google it, these are the leading autoimmune diabetes mouse model. So
who knew there were so many, fancy models of mice?
Scientists are working, which is great.
You get a catalog?
It's not ours. We did not develop it. It's there for a very long while. But we tested the drug on this mouse strain. And again, these mice recovered gaining full insulin independence, which is great.
That is great.
But again, the question is about the infusion. We couldn't pull the infusion out because it's under the skin.
Right.
So we don't know, again,
long term, how much treatment you'll need.
But we know that we were able to, the regeneration process was at least faster than the autoimmune process.
That's very important. Otherwise it wouldn't work.
Exactly.
You're fighting against something that's faster than you, it's not going to work. So that's wonderful to hear. And it sounds like the fact that you did your doctoral work in computational mathematics and on the embryonic cells really was relevant in being able to develop this. So
that was the whole purpose. So this is not, an experimental error like you sometimes hear, the invention of penicillin, for example, which was by mistake, right? Ian Fleming forgot a Petri dish. No, it is, we are planning it. I am planning it for 15, 16 years.
Well, you're a mathematician. So, and I mean that in a complimentary way. You're very mesudar, very organized. Yeah. You know exactly where you're aiming towards and what you're doing, and that's wonderful that you were able to think ahead to know these were the steps that were necessary to bring you to a place. And you mentioned that you have colleagues at Hebrew University that are helpful. Do you have other colleagues around the world or in Israel who are working on similar things?
So many people work on beta cell regeneration, and there is an argument where these beta cells come from. Many groups, also in Israel and abroad. Some of them claim that they can self-replicate because there is always, even in the progressive stages of the disease, some beta cells still survive around and they can induce beta cell replication of these cells. The leading figure in Israel in that field is Yuval Dor, who is a giant worldwide also in this field. There are people who look for stem cells within the pancreas, which is an ongoing debate whether these cells actually exist in the pancreas. I'm not taking any
position
on that one. I don't know, but the leader in Israel is in Tel Aviv University, Professor Shimon Efrat.
So you're all working on different spaces.
Yeah, different avenues, but
parallel to each other.
Yes.
One's replication, you're working on regeneration, somebody else is working on trying to find stem cells which can turn into, so it's...
And there are also many people who work on in vitro system. The Technion is leading that. We have Shulamit Levenberg there, and...
well, lots of wonderful researchers in Israel, and I'm sure around the world as well. But speaking of that, what brought you back to Israel after you were studying postdoc for 6 years out in California? California's beautiful.
Yes, it is. But we wanted to go back. We wanted to come back. Is it a mixture of Zionism, family ties, social connections? The Israeli society is very unique, as you know very much. It has its advantages and disadvantages, but the community is so strong. And we really wanted to be a part of that. I didn't even look for positions in the US.
Good for you. And Bar Ilan in particular?
Bar Ilan, not in particular. I was hoping to get a position in Israel in general. When I was in the US, I was saying always that I hope to come back to one of the 7 leading universities, and everyone was so impressed or thinking that I'm so arrogant. That I
the 7 leading and only.
Yeah, now we have a few
now,
more.
Yes, you have Reichman.
Yeah,
and Ariel.
Exactly, but back then they weren't.
Right.
And I was trying my best. And 2 things for the benefit of Bar Ilan. First of all, they were the most generous. Their offer was most generous. It was the closest one to hit the ground running. Having a lab ready, having the
resources and
I don't know how to say.
An absorption package.
Okay, an absorption package, which was very generous compared to others that were willing to accept me in Israel. And they were also the fastest ones to act.
And you're also able to live up in Tzfat, which is beautiful, it's an amazing place to be. So we've learned a little bit about your research and hopefully the impact it can have. Tell me a little bit about you. Now you did say for the last 15 years at least you've been directed toward this. Growing up, were you always interested in research?
Apparently, yes. I mean, at 5th grade, my mother bought me a chemistry kit.
Uh-huh.
The plastic one, the simple ones.
I remember
that
those.
you drop.
You drop little things
and then
in.
colors are changing,
Yes.
and then you grow crystals. And I was fascinated by that.
Uh-huh.
So much fascinated. Since then, I knew I wanted to be a chemist. Now, am I a classical chemist? Probably not. Am I a biochemist, maybe computational chemist, maybe computational biologist, whatever. But that was the first stages. And after doing military service, which we have to do, and I took it seriously, I started to study chemistry at the Technion in Israel. Mm-hmm. And then slowly, there is a nice story there, because my PhD mentor was Professor Ehud Keinan, mm-hmm. was very much into computational devices. I, at the beginning, didn't appreciate that.
You liked your droppers.
I wanted something more translational.
Right.
And I tried, really tried, and failed the translational project that I was dealing with. It was in catalytic antibodies. If somebody knows something about it, didn't work completely. So plan B was to dive into that. Mm-hmm. And these are nice devices, playthings that you can activate, and you'll get results, chemistry and everything, but I wanted the translational path again. So I took that with his approval and support to Rami Reshef, then a scientist at the Technion, now he's in Haifa University, who allowed me to translate this math into the embryonic system. And this is how things...
And that's how everything took off. So there's a little bit of your, bacterial growth in a petri dish out of surprise, right? Even for a very organized, directed
in a way,
person.
Yeah, failure.
Failure's good.
Yeah, failure brought me to plan B, and plan B is excellent.
Plan B is excellent. Thank you for being with us. Thank you for the work you're doing. One last question. Where do you see the field of diabetes in 5 or 10 years?
5 or 10... So, medical fields are progressing slow. Mm-hmm. So in 5 years, I don't think that much will happen. Even let's assume that my project is on the fast track to approval and it is working great, and people will, let's say, at least will be insulin independent, okay, and free from hypos, events, and this will take time. All the approvals, phase 1, 2, 3, FDA, all that takes time. So I'm sorry.
So 10 years. So give me 10 years.
10 years, 15 years, maybe we will have a treatment for people with diabetes to either regain insulin dependence, hopefully release themselves from the pumps and from the devices, because these are really burdening issues. They take all the spontaneity out of your life. You have to plan in advance. If you see a snack on the table, you cannot take it unless you inject something. So, and if you're running after the bus,
it's true. It can change.
After you injected insulin, you will get a hypo.
Right, and that's very dangerous, as we said before. So you basically want to allow people to run after ice cream sundaes and not have to worry about it. How's that for a nice scientific summary?
Great. Thank you for wrapping me up.
No, this was wonderful. Thank you so much again for everything you're doing and for joining us today. It was a pleasure to speak with you.
Thank you very much, Sharon. Thank you for giving me this stage.